Welcome to the deep dive. You know, sometimes the biggest transformations start small, really small. Like, how does a single fertilized egg even begin to become, well, us, or any complex animal? It's this journey right from the absolute start that's just full of these dramatic twists. So today, we're diving into that very first chapter. Imagine fertilization's just happened. We got this one activated cell, the zygote, lots of chemistries kicking off inside. But what's next? I mean, we think embryo, we think multicellular, but you have to start somewhere. So we're going to explore that first crucial stage, cleavage, those first rapid cell divisions, and then how do those resulting cells arrange themselves into this thing called a blastula?
Yeah, and what's immediately fascinating when you look at the sources is that this whole process of dividing up that first cell, it's not a one-size-fits-all deal at all. It varies dramatically, and the reasons why it varies are actually pretty surprising. So we'll unpack those different patterns, the mechanics behind them, and really what these initial steps mean for the organism down the line. By the end of this, hopefully, you'll see how that single cell really lays down the foundational blueprint, from one cell to many, ready for the next amazing step.
That's a great point. So our mission is to decode these beginnings, look at the why and the what matters. Okay, let's start right there. Fertilization happens, then bam, cleavage. This is that immediate next step, right? The division of that single zygote into lots of smaller cells. What are they called again?
Blastomeres, that's right. And it's not about growth initially. The embryo doesn't get bigger overall. It just divides that initial volume into smaller and smaller units. It's very visually dramatic, actually. Scientists can watch it happen quite easily in the lab. It's this rapid partitioning.
So if it's so fundamental, this division, does it always look the same across different kinds of life?
That's the key question. And the sources are really clear on this. No, the pattern of cleavage is not the same. It's one of the first places you see this incredible diversity of life asserting itself right at the very beginning. It really sets the stage.
Okay, so that diversity, does it boil down to something basic, like does the whole egg divide or just, part of it?
Essentially, yes. That's the first major branching point. We talk about total or holoblastic cleavage. That's where the division lines go all the way through the entire egg. Then there's incomplete or meroblastic cleavage, where only a portion of the egg cytoplasm actually divides.
And total cleavage, the complete kind, where do we usually see that?
Typically in embryos that don't have a massive amount of yolk, so sea urchins, Amphioxus, frogs fit here, most mammals too. And here's a really critical piece of information from our sources. Dense accumulations of yolk retard cleavage and slows it down.
Right, so yolk isn't just food storage, it's like a physical obstacle.
Exactly. It's an architect. It physically dictates how these first divisions can even happen. It's a constraint that drives developmental patterns. Pretty amazing when you think about it.
Okay, this is where it gets really interesting, I think. The frog egg. You mentioned frogs have total cleavage, but it's a classic example of how this yolk influence plays out, right? They're called moderately telolecithal. What does that mean exactly?
Telolecithal just means the yolk is concentrated at one end, or pole.
Mm.
Moderately means there's a significant amount, but not, the entire egg isn't yolk. So in the frog, you've got this heavy concentration of yolk. In one half, we call that the vegetal hemisphere, usually sits downwards. The other half, the animal hemisphere, has the nucleus and less yolk, and it's typically oriented upwards. This unevenness is key.
And you can literally see the effect of that unevenness in the way it divides.
Oh, absolutely. The first cleavage furrow starts up in the animal hemisphere, the less yolky part, and it moves vertically down, meridionally. But when it hits that dense yolk in the vegetal hemisphere, it slows right down. The yolk physically impedes its progress.
Like trying to cut through something thick and sticky.
Kinda, yeah. And it's so slow that often the second cleavage division starts up in the animal region before the first one has even finished cutting all the way through.
Wow. So the second one starts before the first one's done.
Often, yes. The second one is also vertical, meridional, but at a right angle to the first. Eventually, both of these first two divisions do make it all the way through the yolk, so it is total cleavage.
Okay, total cleavage. But then comes the third one. You said that's the game changer in frogs.
That's the one. It's horizontal, sort of equatorial. But here's the twist. Because of all that yolk concentrated downwards, the division doesn't happen right at the equator, the middle. It gets pushed upwards, displaced towards the animal pole.
So it doesn't cut the embryo into equal top and bottom halves.
Precisely. That displacement is crucial. It means the cells produced in the upper animal region are much smaller than the cells produced in the lower yolky vegetal region.
So the cleavage is total, but it's unequal.
Exactly. Total and unequal. And this continues. You end up with lots of small cells up top the micromeres and fewer, but much larger yolk-filled cells down below the macromeres.
This sets up a difference in cell size right from the third division.
Right from the third division. It's a fundamental asymmetry established super early, all because of that yolk distribution.
So what's the mechanism? Why does the yolk cause that third division to be displaced upwards? Is it just pushing it?
It's a bit more direct than just pushing. Remember, the nucleus is already sitting up in the less yolky animal hemisphere. The mitotic spindle, the machinery that pulls the chromosomes apart and guides the cleavage furrow, forms near the nucleus. So the spindles for that third division are naturally positioned above the actual equator of the cell because the nucleus is above the center of the yolk mass.
Okay, so the machinery is already high up.
Right. So when that horizontal division happens, it naturally occurs closer to the animal pole, dictated by where the spindles are. The yolk directly causes this unequal division by displacing the nucleus and therefore the spindles.
And then these smaller micromeres at the top, they divide faster.
Yes, because they have less yolk to impede the process. The larger yolky macromeres divide much more slowly, so the difference in cell size becomes even more pronounced.
Okay, so the frog is a great example of uneven yolk causing unequal division. What about when the yolk is more evenly spread out? You mentioned Amphioxus?
Right, Amphioxus. It's a primitive chordate, and it also has total cleavage, like the frog. The first few divisions look pretty similar, actually.
But the yolk situation is different.
Very different. Amphioxus has only a small amount of yolk, and crucially, it's distributed pretty evenly throughout the egg. We call that oligolysophil, or isolysophil. Oligo meaning little, iso meaning equal.
So less yolk, and it's spread out.
Yeah.
How does that affect the third cleavage?
Well, because there isn't that big concentration of yolk at one end, the third horizontal cleavage happens much closer to the actual equator. It separates the animal and vegetal regions much more equally. The resulting cells are nearly the same size. Maybe the vegetal ones are slightly larger, but it's far more equal than in the frog.
It really highlights how much the quantity and the distribution of yolk matter.
Absolutely. It's a beautiful comparison.
Now, while we're on patterns, there's another type mentioned in the sources. Bilateral cleavage. Sounds like it's about symmetry.
It is. This is where, during the cleavage process, the cells, the blastomeres, become arranged symmetrically across a plane. Think left and right.
So a left and right side get established really early.
Exactly. A plane of bilateral symmetry emerges. You see this in vertebrates, but also in other groups like cephalopods, squid and octopus, nematodes, roundworms, tunicates. It's like the embryo is already sketching out its fundamental body plan, that left-right mirroring, right there in the first few cell divisions. Quite remarkable.
Okay, so we've seen total cleavage, both equal and unequal, influenced by yolk. What happens at the other extreme, when there's just a ton of yolk?
Right. If moderate yolk retards cleavage, what does a massive amount do? It leads to incomplete cleavage or meroblastic cleavage.
Incomplete meaning the division doesn't go all the way through.
Correct. This happens in what are called strongly telolesical eggs. Think birds, reptiles, many fish like sharks and rays. Here, the yolk is so dense and makes up almost the entire egg volume. All the actual cytoplasm, the non-yolky stuff where the nucleus is, sits like a tiny little cap or disc right on top of this huge yolk mass.
So the divisions only happen in that little cap.
Exactly. The cleavage furrows simply cannot penetrate the dense yolk, so only that small disc of cytoplasm at the animal pole divides. The yolk itself remains undivided. It's like a huge energy reserve the developing cap sits on top of.
That makes sense. The yolk is just too much of a barrier.
Precisely.
And is there another type of incomplete cleavage, something even more specialized?
Yes, there's superficial cleavage, which is really fascinating and typical of insects and many other arthropods.
Superficial, like on the surface?
Kind of. In these eggs called centrolysithal, the yolk is concentrated in the center of the egg. The cytoplasm forms a thin layer around the outside, and there's also a little island of cytoplasm in the middle containing the nucleus.
Okay, yolk in the middle this time. How does that divide?
This is the unique part. The nucleus divides first, many times, without the cytoplasm dividing. So you get lots of nuclei within that central island.
Multiple nuclei, one cell.
Initially, yes. Then, these nuclei, each grabbing a little bit of cytoplasm, migrate outwards from the center towards the surface layer of cytoplasm.
Okay, they move to the edge.
Right.
Right.
So now you have this surface layer that's studded with nuclei. Then the cell membranes form around each of these nuclei at the surface. So you end up with a layer of cells on the outside surrounding the central yolk mass. They're initially still connected to the yolk, which they'll use for food. It's a very different strategy.
Wow, okay. Nuclei divide first, move out, then form cells on the surface. Very different indeed. So across all these patterns, we've gone from one cell to many. Sometimes they form a solid ball of cells initially, right? What's that called?
Yeah, that early solid ball stage is often called a morula, especially in mammals. Looks a bit like a mulberry.
But then the next big step is often forming a hollow structure, the blastula. Why? Why hollow it out? What's the advantage of creating that internal space?
Thinking about the big picture. That cavity, the blasticle, is absolutely critical preparation for the next major stage of development, which is called gastrulation. Gastrulation involves massive cell movements and rearrangements. Cells migrate inwards, fold, and reorganize to form the basic tissue layers. That hollow space, the blasticle, provides the room needed for these complex cellular migrations.
It's like clearing the construction site before you start building the walls.
That's a great analogy. You need that internal space for cells to move into and rearrange themselves to build the fundamental body plan. Without the blasticle, those intricate movements would be impossible.
So how does it happen? How does a solid ball of cells like the morula become hollow? What forces create that blasticle? The sources mention the sea urchin.
Yes, sea urchins are a classic model. Several mechanisms seem to be at play. One important factor is osmosis. The cells pump sodium ions into the spaces between them in the center of the morula. This draws water in osmotically, creating pressure that pushes the cells outward, forming the cavity.
So water pressure inflates it like a balloon.
Partially, yes, but there are other factors too. The cells become more adhesive to each other side to side, forming a tight epithelial layer. They also adhere to an outer protein coat, the hyaline layer. As the cells continue to divide, they remain stuck together and attached to this outer layer. Since they can only really expand outwards due to the hyaline layer and their tight connections, this also contributes to pushing them out and enlarging the central cavity. It's a combination of water pressure and structural constraints.
Okay, so it's not just one thing. It's a mix of osmotic pressure and how the cells stick together into that outer layer.
Exactly. Multiple forces working together to create that essential space.
And just like cleavage patterns varied, I assume blastulas don't all look the same either?
Not at all. The structure of the blastula is often a direct consequence of the preceding cleavage pattern, especially the yolk distribution.
So what are the common forms?
Well, the simplest form is just a hollow sphere of cells with the blastical cavity right in the middle. You see this in organisms with little, evenly distributed yolk, like the sea urchin and Amphioxus. Their cleavage was pretty equal, leading to a symmetrical blastula.
Makes sense. And the frog, with its unequal cleavage.
In the frog, because the vegetal cells are so much larger and yolky, the blasticle gets squished upwards. It's not central. It's displaced towards the animal pole, sitting above those large vegetal cells. The floor of the blasticle is thick with yolk cells. The roof is thin with micromeres.
Again, the yolk is still shaping things.
Still playing a major role, absolutely.
Okay, so what about those really yolky eggs? The birds, the reptiles, where cleavage is incomplete. Just that cap of cells on top. How do they form a blastula?
Right, they form a blastula-like stage. The dividing cap of cells is called the blastodisc. The cavity doesn't form within the blastodisc itself, nor within the yolk. Instead, a space opens up between the blastodisc and the underlying uncleaved yolk mass.
Ah, so the cavity is underneath the embryo proper.
Exactly. It's sometimes called a subgerminal cavity. Because it's not truly within the embryonic cell mass, some researchers debate whether it's a true blasticle, but functionally, it's a space that allows for cell movements later on.
A functional cavity, but structurally different. Got it.
It's a modification driven by that massive amount of yolk.
And finally, to complete the picture, insects. Superficial cleavage. Cells forming on the outside around a central yolk core. Do they get a blasticle?
This is the most extreme variation, really. In insects, typically no blasticle cavity appears at all.
No cavity.
None. The cells form that layer around the periphery, and the entire center remains filled with the dense yolk.
So how do they manage gastrulation later?
They have different mechanisms for cell rearrangement that don't require a large internal fluid-filled space in the same way. But from a structural standpoint, you could conceptually think of their blastula stage as having its potential cavity completely filled with yolk.
Wow. So the yolk dictates everything from the pattern of division to whether you even get a hollow space inside.
It truly does. It's incredible how this one component, the nutrient reserve, has such profound architectural consequences for the earliest stages of life.
So let's wrap this part up. What's the big takeaway for you, our listener? We've seen that yolk isn't just passive food. It's an active player. It physically controls how, where, and even if cells divide in the early embryo. It dictates the speed, the symmetry, the completeness of cleavage, and ultimately, it shapes the very form of the blastula, whether it's a perfect sphere, an offset cavity, a cap over yolk, or just a layer of cells around a yolky core. Yolk is the master sculptor here.
Absolutely. Which leaves us right at the threshold of the next big question, doesn't it? We've got this ball or cap or layer of cells, the blastula. It's multicellular, it has some organization, but it still doesn't look much like a frog or a bird or an insect. So what happens next? What are these incredible processes of folding, tucking, migrating the cellular choreography of gastrulation that will start transforming this relatively simple structure into something with a recognizable body plan? That journey from blastula to a structured embryo is arguably even more visually stunning.
Definitely. It builds directly on the foundations laid down by Cleavage, the foundations we've explored today. We really hope this deep dive has given you a new appreciation for those amazing, almost invisible first steps of development, and maybe sparked your curiosity about what happens next in this incredible story of how life takes shape.